Cool. All right. Hi, everyone. Thanks so much for registering and attending this month's Funk Lecture. We're super excited to have Brian McGill as our speaker. Before we begin with the talk, I got a few announcements I want to make about the Art House Conference. So if you submitted an abstract, you should be notified fairly soon or within the week about whether or not your abstract was accepted as either a talk or a poster presentation. And then also this week, conference registration opens up again and with early bird fees going until October 31st and standard registration going on from November onwards. So, yeah, I would try and register for the end of October and save a little bit of money. Um, yeah. And then for all our Flunk lectures, we'll have a Q&A. So if you want to type out a question, we will read it aloud to our speaker, or you can raise your hand and we'll unmute you and you can directly ask your question. And then, yeah, thank you again so much for attending. All of our talks are recorded and uploaded to our YouTube channel. So if you want to revisit this, or if you know someone who wanted to make the talk but couldn't, um, uh, this will be uploaded to our YouTube channel probably within a week. So if you haven't already, you should subscribe and hit the notification button at International Biogeography. Um, yeah, and then for our October Funk Lecture, we'll have Dr. Brett Schaefers of University of Florida give a talk, and I will pass this to Mara to introduce our speaker.
Thank you so much, Raymond. Welcome back, everyone. Welcome back from our August hiatus, and we are coming back with a bang. We are very excited to have Dr. Brian McGill with us here to give what I'm sure will be a fantastic funk lecture. Dr. Brian McGill completed his PhD in ecology and evolutionary biology at the University of Arizona in 2003 and has been a professor at the University of Maine since 2010. And his research focuses on biodiversity, global change, and big data analytics in general. I'm sure that everyone in the audience has read at least one paper by Brian. There's no shortage of those. And throughout his career, the study of species geographic ranges has kind of been a thread running through a lot of the work that Brian has done. But in his own words, it remains very much a hill yet to be conquered. So today we will hear from Brian about his thoughts on species ranges in general, and perhaps also how he actually plans to conquer that huge hill over the next several years. So Brian, thank you very much for agreeing to come and give a talk for Funk Lecture. We are very excited to have you here, and the floor is yours.
Great. Thank you. Thank you to Raymond and Marta for organizing this. Just get my slides up here. And I also am honored to be speaking in the Funk Lecture. I only got to interact with Vicky through IBS, but so this is a very appropriate forum to be, I'm honored to be speaking in, and I hope to see lots of you at the conference in January. So yeah, I want to talk about species ranges. Species ranges are a big concept in biogeography, and sometimes we treat them as very simple, and sometimes they can be found to be very complicated. So I have a lot to get through. I've shared my slides so people can download the slides if they want to follow up on references. About half of what I talk about is going to be kind of well established in the literature. And I'm going to go through that pretty quickly. And about half of what I'm going to talk about is recent research, mostly with myself or collaborators. So species ranges, as I like to point out, is something that even non-scientists know what they are. They exist in every bird book, every tree book. On the right-hand side at Wikipedia, almost every species page in Wikipedia has a species range page these days too. They sometimes call them the blob in a guidebook, and everybody knows exactly what I'm talking about. So on that level, they're extremely widespread and understood and valued. Species ranges have been a focus of a lot of study. At least two whole books have been written on species ranges, and they have been key chapters in a number of books on macroecology as well. And species ranges are pivotal in lots of applied questions as well. Climate change is, to a large degree, a focus on species range shifts, at least as new species come in and species leave. Those are shifting species ranges. This, of course, is past change with the glacial retreat. And species ranges are basically the story with invasive species too, right? They get to a new continent and then they re-expand out and fill a new species range. So there's lots of interesting natural experiments there, but also lots of applied questions. And in a conservation context, for red listing, criteria B is the size of the range. In particular, having a small range can get a species listed, and that's one of 5 criteria. So it's a very pivotal concept. Real quickly, the data sources that we use to build species ranges can be presence only, presence absence, or abundance-based data. And they often are point samples, but they sometimes are rasters. The rasters, of course, at some point probably came from point sampling, but sometimes it's easier to work with raster level data. And there are all kinds of techniques. I'm not going to spend a lot of time talking about them to delimit species ranges. The simplest is to just take, for example, latitudinal limits, 2 lines, and what's the range of latitude. Latitude, longitude boxes, the red lines. The convex hull is one of the most popular techniques, which is just taking the boundary we can draw that's convex. Alpha hole ranges kind of take a convex hole and eat into it to try and avoid really obvious regions of the convex hole that shouldn't be included. There are more sophisticated methods based on contouring and 95% kernel density estimates and things like that. There are expert ranges. This is longleaf pine, and this is Little's expert range shown in green. And of course, in recent years, we've had species distribution models where you're basically leveraging climate data to try and interpolate a species range. That presents a problem with thresholding, which is not a trivial problem in itself if you actually want to draw a species range. But these are all techniques that we use. Good news is there's a pretty high correlation in terms of range size between all of these techniques, but of course, there's lots of difference in there too. Just to note, range boundaries are not as precise as they look when we draw them on a large scale map. They're always a bit fuzzy. There's a petering out that happens. You see a conceptual diagram on the left, the example of a tree line, and that one's actually probably more of a sharp tree line than you often find. And just to note on the right, a personal story. When my son was little and had colic, I spent a lot of time driving on the freeway, which was basically a transect through the edge of the saguaro range. And I identified the very last saguaro. And it was a real story of microclimate. It was in a wash down in a canyon, so protected from winds, but it was also up on a little hill inside of the wash. So it was not getting the cold pooling at the bottom of the wash. It was just, to me, an interesting example of how microclimate is part of this story, very much of species ranges as well. It's quite well known. Species ranges, the total size, if you look at area, or even if you just looked at lat long limits, is highly skewed on an arithmetic scale. It's pretty close to log normal, but like body size, it actually seems to have a little bit of left skew from a pure log normal distribution. Over on the left is Birds of the World by Orms. Over on the right is a figure from Gaston looking at several different groups on an arithmetic scale and several different measurement methods. It's really just like abundance and body mass being log normal. Or approximately log normal. It's an extremely reliable pattern. Range sizes, like abundance, are not phylogenetically conserved. They change very quickly across the phylogenetic tree. Just a simple example, these are they're not sister species, but they're congenerics. Used to be Dendroica, now I think it's Cetophaga. The North American warblers or New World warblers, and Kirtland's warbler, but there are only like 30 species in the genera, so these are pretty closely related species. Kirtland's warbler just covers this little tiny tip that's blown up in an arrow. Maybe its range has expanded under recent conservation efforts, but for a long time its range was just this little tiny pinprick in Michigan. And yellow warbler covered large parts of northern US and Canada. And it's not super obvious why. Like I say, they're closely related. One's a 14-gram insect gleaner. The other is a 16-gram insect gleaner. Very similar niches, very similar nests, very similar mating behaviors. Probably the biggest difference is habitat specificity. The Kirtland warbler lives on the ground and only lives in jack pine forests that have had recent burning. And yellow warbler is a habitat generalist. It will live in both boreal and deciduous forest. But, that kind of gives you the idea of the range of species ranges. Some well-known rules. Rappaport's rule says the species ranges are bigger at high latitudes, far from the equator. That by and large seems to be holding true, although there are certainly some specificities in taxa and location. Again, the birds of the world from orms, it looks much stronger in Eurasia than it does in North America, although it exists in North America. Another paper by Naya Morita Holmes looking at tree ranges. It appears like there's a secondary large range region in the Amazon, but that's also, if you control for species diversity, that disappears, and it really does look like the large ranges are fairly far north. Another pattern is range size and average abundance are strongly correlated. This is just a quick graph from the breeding bird survey that I put together. So an obvious question, what creates range boundaries? There are very well studied examples where climate is an extremely strong predictor. On the left is an example from Terry Root, Eastern Phoebe. If you look at the -4 degree minimum temperature isocline and the edge of the range, that's a pretty strong concordance. Another example, marine example, fiddler crabs. Their larvae seem to need a temperature of at least 18 degrees Celsius to develop in the summer, and that seems to be setting the range boundary. This is 18 years old. It'd actually be interesting to go back and follow that up. I don't know if that's been done, but that seemed to have set the range boundary quite strongly, even though there was pretty good gene flow. Sometimes it's competition. On the left, this is also an example from Terry Root, red-headed versus the red-bellied woodpecker. Their ranges largely overlap, basically the eastern U.S., contiguous U.S., but their abundances, I don't know if you can see the mouse, but are extremely different. I think it's the red-bellied woodpecker is much more abundant in the south, and the other woodpecker is much more abundant in Missouri. So even though the ranges strongly overlap, their abundances are clearly shifting from competition. This is a famous example from Diamond, where on elevational ranges, and the key test here that's strongly implicating climate is when a species disappears, the other two species or one species expands to occupy the full range, but when both species are present, there's a range separation and a range boundary. Root has done, I'm not sure I've seen another update to this. There probably is that I'm missing and forgetting, so my apologies, but Root did a big analysis with birds in 1988, and this was her story, northern ranges versus eastern versus western ranges, temperature, frost, vegetation, precipitation, elevation, and humidity. So basically vegetation, habitat, and temperature both played strong stories. There's this claim that's pretty widely believed in. I'm not sure I've ever seen a really strong test of it. I call it the MacArthur-Dobzhansky hypothesis because both of those early biogeographers talked about it, which is that the poleward in the northern hemisphere, the northern range limit is set by climate, and the more equatorial or southern in the northern hemisphere range limit is set by competition. It's a compelling theory, but I'm not aware of a really robust, many taxa test of that. As a result, because there are many factors setting range limits when ranges move, this is by Kate Lyons looking at mammal ranges from the last glacial maximum to the present time, or recent past time, 500 years ago, I think. There were substantial range shifts. The average species shifted 1,300 kilometers, but what I find most interesting is the direction of the range shift. There's some bias. There are only a couple of species shifted due south at 180 degrees, and there's some bias of kind of north by northwest shifting being most common, but there's an incredibly diverse range of directions of shift. Even as strong climate change, 5 to 7 degrees Celsius of global warming, glaciers retreating northward, you would think the story would be the strong northward shifting, but it's not. It's actually a pretty diverse directions of shifting, which I think is a direct function of the fact that the range boundaries are so complicated. I'm going to give you a worked example. This is in prep with Daniel Lachlan. And looking at the saguaro, just because it's a well-known species, it's also a species people think they know what sets the range boundaries quite well. So over on the left, I have a plot of mean minimum temperature in the winter, and I have the percentage of the rainfall that happens in the summer. And you can see this red line here actually sets a very sharp boundary. It's a little bit sloped, so it's not entirely percentage of rainfall, but where the species is found, there's there's a little pocket that's beyond, which corresponds to this little pocket over here. But what's been known for a long time is the northern and eastern range limits as you go up in elevation are strongly set by this minimum temperature. And in particular, it's quite specific. They can't tolerate 36 hours of freezing. They can tolerate one night of freezing, but not a day and a night of freezing. So that sets that range boundary. And the southern range boundary has been pretty widely understood to be set by competition. I'm using NDVI here as a proxy for competition, but there are a bunch of specific species, some larger columnar cacti, and starting to get some trees that set the lower range boundary. So in this diagram, the yellow is the intersection of all these factors, and the green shows the extension for just a single variable. But if you take the intersection of all of these, you get the yellow range. It works pretty well with just 4 variables, but there are some clear exceptions. There's a region over here. The Rocky Mountains are in the way, and also the fraction of winter rains is probably too low over there, which is not shown. The big mystery is what sets the boundary here. It very closely follows the Colorado River boundary between Arizona and California. But but as much as people have tried, and including I tried pretty hard to find a specific climate limiting boundary, it doesn't exist. And there's actually this pocket over in the Superstition Mountains. Some of these are in Palm Springs, California. They're clearly transplants, but some of these are natural species, native species, far separated. Almost feels like a dispersal limitation boundary with the river, but they grow on the west bank of so it's kind of a mystery even what sets that. The other 3 boundaries are pretty clearly set. That's just a case study. One of the big issues with looking at range boundaries is there's pretty strong evidence. The evolutionary folks have produced very strong evidence for local adaptations. So there becomes an evolutionary, we can look at the ecological boundaries, but there becomes an evolutionary question of why species don't just evolve to expand their range boundaries. This is a nice example of local adaptation. So species from the blue region grow the best when planted in the blue northern region. Species from the intermediate purple region grow the best when planted in the intermediate purple region, and species from the south grow the best when planted in the south. Notice there's also these nice curves where regardless of where planted, the blue species grows the worst but is the most tolerant, and the red species from the south has the least narrowest niche but has the highest fitness. On the other hand, it's not very hard to find figures like this from Schultz with tiger beetles, where the preferred temperature, the temperature that if you give a beetle time and follow it around to see where its preferred temperature is almost exactly the same across Arizona, Colorado, Maine, and Wisconsin. So a very large fraction of the contiguous US. So there's an evolutionary question hanging over this. I don't know that anybody has a good handle on that. There's a strong assumption that ranges are equal to niches. This has been called Hutchinson's duality. There's a nice paper by Rob Colwell and Thiago Rengel, but it's very clearly laid out in the Hutchinson 1957 paper. That's actually, he's famous for fundamental and realized niche, but really what Hutchinson's in that paper is doing is setting out this duality between geographic space and environmental space, which now allows us to take geographic distribution and turn it into niche distribution. One of the best ways to test that hypothesis is reciprocal transplants. You take a species and you grow it inside the range and outside the range. 2 nice review papers by Hargreaves et al. And Lee et al. They broadly found support for the notion that range boundaries and niche limits co-occur. It was far more common that fitness declined outside of the range than inside the range. But I find especially interesting the Hargreaves test. They did an analysis where there were 3 plants, three populations in the interior, the edge, and the exterior of the range. And they actually specifically compared fitness to one, right? So it'd actually be a boundary of distribution. And what they found is they only had 6 data points, but for geographical ranges, the niche was larger than the range. In other words, the population outside of the range did well 83% of the time. So I think it's an open question how well ranges really map to niches still. I guess I'd highlight that as an open question. Probably the best developed theory to date on range boundaries is Soberone and Peterson, also in their book. The band theory, which is expanding on Hutchinson and talking about biotic, abiotic, and movement or dispersal limitations to niches. And of course, the realized niche, where a species is actually observed, is just right in here where the species is able to get to and biotically and abiotically can coexist. The big question then is how are these niches related, right? This is just a Venn diagram drawn for convenience to expose all the different regions, but there are different theories and they cover this where you might imagine the niches are strongly concordant, or the biotic niche might be very different from the abiotic niche, or the movement niche might be very different. So the Hutchinson model, and I think Alan and Jorge have done a good job of extending, describing that model. And I think they are the first to recognize these limits, but there's real limits in this approach. First of all, the limits are not binary, right? There's more or less, when you talk about biotic or abiotic, there's more, even dispersal. There is variation, right? May get there once every million years or might get there once every 2 years, and that's a big difference. And biotically and abiotically, fitness could be very high or it could just be above 1. And these filters interact with each other and they're highly non-sequential. We like to treat them as sequential. Does the species get there? Can it survive abiotically? Then can it survive biotically? But of course, the fitness, these all interact with each other. And one example of that, this is just using geographically weighted regression, which allows the coefficients to vary across space. And this is a bird species, the dickcissel. I'll show you this data several times. But this is the limiting factors as they vary across. So here's the actual plot of abundance. But what's limiting that abundance varies very much across space. In some regions, it's precipitation. As you move west and it gets drier. In other places, it's minimum temperatures or maximum temperatures. Some places just having enough warmth in the growing season. So there's a strong non-stationarity, and we often ignore that non-stationarity in space of the controlling or limiting factors. And this is just illustrating this notion that it's not a binary limitation, right? So this is going back to Birch's flower beetles and Park's data in the 1950s, 2 species of beetle. And this is a plot of fitness, right? So lambda equals 1. This is a plot of fitness versus humidity and temperature. And it's not a binary circle. And that really matters when you put 2 species together. The range of both species is much larger than where they're found in competition. And so the competitive outcome is conditional on climate. I think everybody knows that intuitively. But once we start talking about species ranges and niches, we often forget that. Here's a paper with Daniel Laughlin. And we were really interested in the notion. So we are kind of making the Hutchinson duality assumption that we can go from geographic position to niche position, which I think, especially in adult trees, is probably not a terrible point. But we highlighted a pretty long-standing literature that what you see in nature, which is what you see down here at the bottom, is that the niches look distinct. When you go out and you observe where species are actually found, the niches tend to be maybe slightly overlapping, but they show this nice gradient. But the realized and potential niches that create this structure, there are multiple possibilities that all create this same identical structure. So the traditional assumption is that the potential niche, using that as sort of a more precise version of the fundamental niche, but I'll describe how we got that potential niche in just a minute, is really very closely related to the realized niche, and species really are organized across an environmental gradient here, mean temperature. But there's an alternative model known as the shared preference niche, which, and this looks exactly by not by coincidence, the local adaptation data I showed you. One species is nowhere maximally fit, but has a very high environmental tolerance. And this other species, the brown species, has maximum competitive ability and wins competitive battles, but has lower environmental tolerance. And the point is, the potential niches look like the lines here, but the realized niches look like these areas between the pluses, which is exactly the same over here. And there's a slightly different version, which is centrifugal, which is where the potential niches, sorry, the realized niches lean, no, sorry, the potential niches lean into the center, but the realized niches, again, are distinct. So we did this with trees, and we looked at arboreta. We took arboreta records as a sign of where the potential niche was. If there's an adult tree in an arboretum, it's a little iffy because there's probably some watering of seedlings and things like that. But the point is dispersal limitation is removed. And if there's an adult healthy tree, we took that as the potential niche and we compared that to the realized niche using data like the US Forest Inventory. And what we found was actually very strong evidence for the centrifugal niche. So colder species found further north, their realized niche was, of course, more northerly and colder, but their potential niche stretched towards warmer temperatures. And conversely, very southerly species, warmer niches, their realized niches were warmer, but their potential niches actually stretched towards colder. So this is an example of a centrifugal, and we actually found that almost every tree species potential niche covered 12 degrees Celsius. And we found that in general, the realized niche was less than 75% of the potential niche. This idea has been around for a long time. Colwell and Fuentes put this idea in 1975. Mike Rosenzweig's put it out in multiple papers. There's a nice experiment by Campbell and Grime in 1992, which I include in the community ecology textbook with Gary Mittelbach. Irene Wishu did a survey, a review of papers in 1998 and found that for most cases, this shared or inclusive niche paradigm was what was found. Fetus found this in rhododendrons up the side of the Himalayas. Breffelt did this with the large common garden experiments for forestry species and generally found that a species grew best at a warmer temperature than the temperature that it actually came from. Not species, but genotype grew best at a warmer temperature than where the genotype was collected. Okay, so that's looking at ranges as whole cohesive units, although with my points about interactions between variables and not having binary boundaries, I've kind of made a pretty strong, hopefully in the non-stationarity, geographically weighted regression, hopefully made a pretty strong argument that it's a mistake to just think about the edge of the boundary and we should think about what's going on inside the range. We've known for at least a couple of decades that there's very strong, if you look at a single species and then you look at the abundance of that species across its range, there is once again log normal distribution. There's a highly left skewed distribution, or if you plot it on a log scale, you get this kind of log normal distribution with a bit of left skew, meaning that the species is rare almost everywhere. This is kind of something people tend to talk about species that are common or rare, but when you actually look across its entire range, every species is rare in most of its range. What, the real action and where we think of it as a common species is that small area where it's most common for it relative to its range. And, those abundances might be, for some bird species, might be higher or lower. So, sorry, went backwards here. These abundances are reasonably consistent through space and time. In other words, there is spatial autocorrelation. Or here I've just done correlation between nearest neighbor points in the breeding bird survey. The correlations are 0.6, 0.7. So there's a pretty strong, you can also just look at autocorrelation, Moran's index. There's a pretty strong autocorrelation. These ranges are also pretty strongly correlated through time. There's a correlation of 0.7, 20 years apart in the breeding bird survey. This is a smaller scale study within Tucson, the Tucson bird count. It's a little bit noisier, but there's still a positive correlation. So it makes sense to talk about abundant surfaces. These abundances are not just randomly bouncing around chaotically or something. There actually is spatial structure that's persistent through time in these abundance structures. This is a classic figure by, oh, I left the name off, but it shows a species distribution. And so here's the whole range. Here's a region. Here it grows on limestone outcrops. Here's the cluster. Here's a glade, and here's individual plants within a glade. So the point being that there's a strong, no matter what scale you look at, there's a strong clustering. Species or clusters of species tend to occur near other individuals or other clusters of individuals. So it's a highly this is a spatial version of, right? And all this empty space is that long left skewed tail of low abundance. So it's a more spatially structured version of saying abundance is log normal across the species range. People have gone further and commented that within a geographic range, there's lots of holes where a species is just absent. Rappaport called it the Gruyere cheese model. Hurlburt and White, bringing the North American perspective, called it the Swiss cheese model. Those, of course, are very similar cheeses that have the big bubbles in them. And Svenning and Skov talked about how ranges were, the filling was limited. They identified 38% filling. They attributed this to possible climate disequilibrium. Ben Seliger, working back in the paleo data, found that there is a consistent underfilling of ranges as well. So these ranges are very much not a solid entity, but they're actually huge holes, and they're also strong peaks. I had my one chapter unpublished for my dissertation. I went and identified what I call the peak and tail pattern. I won't go into a lot of detail. The peak is abundances that are 80% of the maximum observed abundance across a range. And I think the tail was anything that's below 20% of the maximum observed abundance across the range. So each of these vertical lines is a species. The black are the proportion of sites that are above that 80% of the peak. The white are zeros, completely absent. The grays are below, they're in the tail, they're below 20% of the peak. And the darker grays are these intermediate transition zones, right? If you think about a Gaussian bell curve, which is a stereotype of abundance across a range, there are these shoulders where it rises from the tails up to the peaks. So the dark gray, but you can see the peaks are very small, about 6%. The tails are quite large. They're over, including the non-zero and the very low abundance parts, are about 60 to 70% of a range. Again, it's another restatement of this log normal distribution, but suggesting there's some spatial structuring. People love to keep assuming the peak of abundance is in the center of the range. It's just not true. Austin commented on this in 1985, Blackburn in 99, Seguin and Gaines in 2002, Tad Dallas, a couple papers, Luca Santini, a couple papers. Part of my unpublished chapter, I looked at the relative abundance, so compared within a species. I divided the range into 4 concentric rings. They weren't exactly rings because ranges aren't circles, but kind of working inwards from the convex hull. And the average abundance in each of those rings was exactly, well, very, very close to 1. I guess the dashed line is the actual observed relative abundance, and the solid line is the observed abundance. So the abundance, and this is averaging across many species. So in one species, the peak might be out at the edge, and another species, the peak might be in the center, but averaging across species, there's no preference for being peak abundances being at the edge or the center. I'm going to show you some actual abundance surfaces. So here's the dickcissel I've talked about. And here's a transect across the range. The blue dots are what are actually observed. This green line is trying to fit the stereotypical Gaussian bell curve through that data. The dickcissel is always shown because it actually comes pretty close to fitting the Gaussian bell curve across its range. It also comes pretty close to fitting that the peak is in the center. But I'm going to show you some others. Here's the scissor-tailed flycatcher. A couple things going on. It clearly has 2 peaks, neither of which are central. This is a more extreme example, the canyon wren. It's probably still got 2 peaks, not coincidentally in locations where the geology has lots of canyons. But the range is much, much larger than that. Here's a raptor, Cooper's hawk. It's got a very large range, but this begins to look almost like an eruptive model. I'm not aware of anybody who's figured out why these spots show up where they show up. And it's pretty hard to say there's one peak. There's arguably, maybe this is a peak, but there's arguably dozens of peaks. I call this a more eruptive model as opposed to a peak and tail model. Here's the red-bellied woodpecker that I was mentioning earlier, which the range covers most of the eastern contiguous U.S., but it's concentrated in the southern U.S., southeastern U.S. in terms of abundance. And so clearly not a central abundance, but if you take a transect, it's not a terrible fit to a Gaussian bell curve kind of idea. So the peak and tail, that's birds. These patterns do generalize across other species. I don't have time to show you insects and other things, but here's data from the U.S. Forest Inventory as assembled by Iverson. And you can see the same notion. Here's the hemlock. Its range covers a pretty large chunk, but its concentration is clearly close to where I am here in Massachusetts and Maine. White oak, again, covers the whole eastern U.S., but its concentration is closer to the forest prairie transition where it's drier out west. So it's quite everything I've been showing you mostly has come from birds because we have the data, but they're quite general patterns. I have yet to see them contradicted in a general sense across many species in other data sets. So to summarize, abundance across a range does show spatial coherence. It makes sense to talk about abundant surfaces. And by and large, there are some species that show that eruption pattern, but most species show what I call a peak and tail pattern. There are 1 to 3 peaks with very high abundances, orders of magnitude higher than the tails. And typically 1 to 3 peaks, they're quite small. They're very concentrated peaks, about 6% of the range. The tail, which is 0 or near 0, covers most of the range, about 65%. They're small transition zones. The peaks they're randomly located. They're not necessarily in the center. And the peaks are orders of magnitude more abundant than the tails. Okay, so that's a geographic. What if we switch to that Hutchinson duality and think about across climate? If you take data and you plot abundance versus any climatic variable, this is very typical of what you get. You get something that does look like a bell curve, which is why I call this the Gauss biogeographic law, but it also looks like this bell curve is an upper limit. You can think of quantile regression. This is a nonlinear quantile regression. And most sites, so again, this is one species, many sites, each of the dots is a site. Most sites, the species is below that potential peak abundance for 5,000 degrees growing degree days, which is why I call it the Gauss-Liebig law. Liebig talked about the one limiting factor. And I first discovered this in a paper with Stacy Jerema, Jason Sampson, and Murray Humphreys, who had beaver data. They kept plotting this data. They were trying to predict abundance because it was a wildlife context, and they kept getting really messy plots. And I finally realized that's actually a feature of the data rather than a bug, that it has that noise because these are really quantile regression plots. This is the Dick system again. This is a bunch of different climatic variables. And I don't think I didn't get the figure in, but the point is that, the species that is at a potentially optimum growing degrees of 5,000, but is way down here with a low abundance, that's because it's over in the tail here of, I don't know if it's this particular climatic variable, but it's in the tail of another climatic variable. So it's a kind of a multifactorial view of climate where the most limiting climatic factor drives the abundance, and that causes most things to be close to 0. But where all of the climatic variables come together at their optimum, you can get these higher abundances. Again, you can get similar patterns in trees. So this is not something unique to birds. And here is where you can actually go through and say, well, which of those climatic variables is the limiting factor that's setting the abundance? Where is it hitting that 90th percentile contour? And it actually works surprisingly well. This goes back to the notion of the geographically weighted regression and non-stationarity. This is the Dick system again. It's most abundant right here. But in this northern area, the mean temperature, just having enough warmth to get through the growing cycle is important. Over here, the summer maximum temperatures are important. And when you go further south, it's the annual variability in precipitation, basically getting enough summer precipitation to have the productivity to be able to grow. So you can actually, take that Gauss-Liebig law and start to combine that with the idea of non-stationarity and describe different limiting factors across a range. Okay, what I'm going to show you now, it's once again the Dick-Sissel, but I'm going to show you, I think it's 5 years of data. So I just want to orient you. Color scale, so abundance of up to 300 individuals, 350 individuals on a 25-mile, roughly 40-kilometer route. And so here's this peak of high abundance. Out here, the black dots are not zero, but they're very close to zero. And the open circles are inside of the range according to a convex hole, but they were measured as zero in this year. So I'm just going to scroll through several years of data. And I don't know what you see, but what I see is the peak stays very constant. It shows some change, but not a lot of change. And out here in the tail, it's kind of bubbling. It's the changes in abundance are not necessarily large. Oops, sorry. It's maybe going from 0 to 2 to 0 again, and out here, maybe going from 300 to 250. It's actually might be a bigger change, but as a proportional change, it's not so big. That the variations out in the tails. And you can actually confirm that. This is fitting the Ricker population dynamic model. So this is basically B. It's the exponential lambda, the population growth rate, plotted across space. This is C, the density-dependent parameter, and this is V, the variance in the population. And here again is the abundance. The correlation between the population growth rate and the actual abundance, so the y-axis here is abundance, it's actually slightly negative. It's basically zero. The density dependence factor has a pretty strong relationship. And actually the variance has a pretty strong relationship to the abundance as well. In a Ricker model, the equilibrium population size is log B over C. So it's not surprising that this 1 over C relationship has quite a strong relationship. It is pretty shocking that the log B population growth rate shows almost no relationship to abundance. This was my postdoc, was to look at population dynamics in this fashion using the breeding bird survey across space. And I assumed, like that hundreds of literature before me, that all the action would be here in the population growth rate. And we'd find this nice correlation between population growth rate and the peaks and tails of a species range. And we didn't. So I've spent 15, 20 years actually trying to figure out what's going on. So if you do this across all species, the average correlation between lambda, which is log B, and abundance is actually negative. So where it's more abundant, it grows worse. The variance is negative, but that means the coefficient of variation. Well, yeah, so low abundance populations have a higher variation. And if you turn that into coefficient of variation, it's even stronger. And this density dependent term does have a strong negative effect. The best three growth rates. So I started looking at the variability because the variability correlation was the strongest. And the top three growth rates, again, were negatively correlated. They showed a similar relation to the average growth rate. But my kind of aha moment was when I looked at the three worst growth rates across a 20-year period. And that's actually positively correlated with abundance. So that suggests abundance is really a story about bed hedging and not having really bad years. So if you're going to be super abundant, you can't ever have a really bad year. And so I don't have too much time to spend on this, but I went overboard and I looked at dozens of variables. I fit the Ricker model. I fit the logistic model. I looked at these more empirical measures like the single worst growth year and the single best growth year, the average growth rate, the actual variance, the variance estimated from the Ricker model, the coefficient of variance. And I ended up doing a principal component analysis of these variables across the geographic range, and then looking at how they're correlated with abundance. And this pattern with the Dick-Sissel is pretty typical. The first principal component, the strongest correlations are positively abundance. Again, this is abundance across space is what I'm trying to explain. Positively correlated with the lowest growth rates. So if you have a very high low growth rate, you'll have a high abundance. And they're actually very negatively correlated with the peak growth rates and negatively correlated also with things like lambda, the population growth rate, or the logistic R growth rate. They're negatively correlated. So the bed hedging principal component is the primary explanation of variation in population dynamics across space, and it's also the primary variation in explaining abundance across space. The second axis came out to basically an R or lambda, and the third axis came out to basically a K or a carrying a C, because it's an inverse relationship with C for carrying capacity. Basically, the second two axes came out almost like you would expect, R and K, but the first axis is very much a bed hedging. This kind of repeats across different bird species. So yeah, coming in right on time here. I know I talk fast, but feel free to download the slides. To summarize the talk, ranges are pretty fundamental, even to non-scientists, but they're not very easy to actually define and draw the boundaries. The variation in range size is log normal and quite consistently found and well understood. To me, the log also empirically, when people have gone out and looked at correlates with range boundaries, it's sometimes temperature, it's sometimes precipitation, sometimes habitat, it's sometimes competition. So range boundaries seem to be a complex mix. I would kind of suggest this log normality of range size tells me that it's a central limit theorem kind of a combination of many, many factors. And it may be hard outside of a case study like I did with the saguaro to go deeper on a general theory of range limits. Range limits show pretty good correspondence with niche, although I think there's still some interest in the fact that so many species, especially when you look at geographic niches, if you take it and you plant it outside of its range, it still seems to grow well. It's a little bit of a conundrum that is not fully explained. I think the idea of local adaptation is also a conundrum that's not been fully explained. When you compare the potential niche or fundamental niche to the realized niche, they have this strong inclusive or shared preference. And in fact, they have a centrifugal organization. It's very different from the default assumption of, well, the fundamental niche is just take the realized niche and expand it out in both directions a little bit. It's actually saying that they're completely different and that everybody has a potential niche that leans towards this shared environment of about 12 degrees Celsius. But their realized niches, they're getting pushed by competition out to the very distinct temperature gradient that you observe in trees across North America. Abundance within a range has a massive amount of low abundance in holes. There are very few peaks. Their peaks are very small. The peaks are more or less random. In that Gauss-Liebig law, it suggests the location of the peaks also is a many causal multiple factor environment. And so it's not surprising you get this log normal pattern out of it. Abundance across a climatic gradient, as I just mentioned, follows this Gauss-Liebig law, where any given climatic variable sets an upper limit, but doesn't predict the actual abundance because there's many climatic variables interacting. And population dynamics across a range emphasize bed hedging, and bed hedging seems to be the best predictor of where those peaks are occurring. So net, the conclusion of my conclusions, ranges are measurable and show a lot of repeatability, including the abundance structure across a range. Ranges range sizes and locations of peaks follow quite a few laws quite regularly. And in particular, the fundamental or potential versus realized range seems to have this pretty consistent pattern. Edges of range and locations of peaks seem to be very multi-causal. They seem to have this kind of log normal central theorem kind of distribution. So you can tease them out in a case study. I'm not convinced that there's a general theory to be had of those. I do think something that comes out of some of the work I was repeating towards the end is variability. We've always kind of looked at mean temperature, mean precipitation. Not always. Plenty of people have thrown variability in. And indeed, when I throw it into SDMs, the variability often comes out as important or more important than the means. But I think think we need to look at the role of variability in species ranges much more than we have. So I will stop there and stop sharing, and I look forward to your questions.
Thank you very much, Brian. This was fantastic, and so much packed with awesome information. Okay, I already have some questions in the chat, so let me read them out for you. The first one is from Britt Poo. Do you think all of the patterns that have been discussed here would hold if you looked at species data on oceanic islands? If not, how might they differ?
Yeah, it's a good question. I mean, the problem you get into with the islands is how you define the range extent, right? Because you have a bunch of water in between, and and so you can look at larger oceanic islands, and species have ranges within those islands. But in other cases, it's more of a occupancy, presence, absence on an island kind of data set as opposed to a geographic range across a map. A lot of the patterns I talked about range size also occur in occupancy. I mean, that's a topic I didn't have time to get into, but occupancy also tends to have a few regions of very high occupancy and lots of areas of very low occupancy. So I think probably with islands, I would think about it in terms of occupancy, which I didn't get into, but I think it shares a lot of the same story as ranges and range sizes.
Okay, well, thank you. And I think the next question is kind of related from Mark Lomolino. Masterful work, Brian. Much, if not all, relevant data is from two-dimensional ranges, terrestrial species. What patterns do you anticipate to emerge for 3D ranges, marine and aerial species?
Yeah, I mean, it's a great question to add that third dimension. I think in terms of vertical niche, if you think about, for example, in marine species and the depth that they're found at, you tend to find you can do the same thing, right? Species have niches. There's gradients, and things are found at different depths, and they have a nice niche separation. I don't know the marine literature well enough to know if people have shown that you get this kind of inclusive or shared or centrifugal niche organization on a vertical gradient. I kind of suspect you do get a shared preference with everybody wanting to be at the top of the water if they could, and it's only competition that's driving people down. I guess I can think of specific examples I know that look like that, but I don't know how general that pattern is. And vertically, yeah, the vertical niche is more complicated. I mean, if you're a bird, you can be up to several thousand feet, but you're going to spend most of your time in a tree or where the insects are. Be interesting though, for example, with bacteria, when you start to get into the aerial bacteria niches, what those patterns are. I suspect they continue, but that's just a suspicion. I don't know the answer for sure.
Mm-hmm. Okay, and I think the next question is kind of related as well. So let me see if you have something to add to what you've already said. This one comes from Sally Keefe. To what extent do you think, or does evidence suggest, that these patterns apply to marine species? So you kind of touched on this already, but do you have anything else to add?
Yeah, the vertical component I think is an interesting complexity. The horizontal component, by and large, certainly like the log normal distribution of ranges, the peak and tail pattern, all the data that I've seen is those extend into the marine realm quite well as well. You definitely have concentrations of abundance of species in the marine realm. You have very large ranges where the abundance is low. The ranges are distributed log normally. Yeah, so I'll go out on a limb without having shown any data and say that the horizontal two-dimensional patterns are largely very consistent. There's a little bit more mixing, right? And also the sampling in the marine world is more time averaged. So I think those holes probably get time averaged out a little bit more in a marine context, but I don't think it changes the basic story of beaks and tails.
Mm-hmm. Okay. Question from Paulo Borges, and I apologize if I mispronounced your last name. What about elevation ranges on islands with endemic species abundance peaking on pristine habitats?
Peaking on pristine habitats.
Mm-hmm.
Yeah, I mean, we tend to treat elevational ranges and geographic or horizontal ranges as equivalent. And for the most part, I don't have any reason to think they're different, except, of course, the big difference is dispersal, right? It's much easier to disperse up a mountain 1,000 feet than it is to disperse geographically far enough outside of a range to make that noticeable. That could be hundreds of kilometers. So the dispersal, I tend to think, is the one big difference. As far as the peaking in pristine habitats, yeah, there's a whole global change angle that I didn't talk about. One of my reasons for pursuing that Gauss-Liebig law is I was just throwing climate in there, but it would be very interesting to start to throw human impact factors in as the gradients as well. And the result that is being mentioned kind of sounds like you're going to see that same pattern, but I'd love to look at it in that context.
Okay. Richard Field asks, thanks, Brian. Should we be surprised that the general conclusion is that there are multiple limiting factors producing a normal distribution for the very reasons that such a distribution is called normal? It seems to go against conventional thinking about ranges and abundances in various ways, but in retrospect, surely makes sense.
Yeah, I've been having conversations about the log normal distribution with people for 25 years. Some people find it very comfortable and very obvious, and some people just hate it. Partly because I think, there's this drive to get more mechanistic, and to many people, the log normal distribution doesn't feel like a mechanism. To me, I do think it's a mechanism, and when it's showing up, it's maybe a sign to me we shouldn't be looking for deeper, physiological level mechanisms. So, I mean, my answer to Richard is it depends on your perspective. I'm more than happy and not at all surprised to find the log normal in so many places, but I've had conversations with people who tell you that's not a mechanism. It's completely unsatisfying. We have to go deeper. It has to be reduced to population dynamics, or it's not ecology. So, and I'm sure we have both those perspectives represented in the audience here. So
I'm sure that. Okay, next question. I'm gonna go through them quickly because we don't have much time left. But a question from Felipe Espinoza. Do you think the same patterns of abundance can be seen in invasive species ranges?
In terms of the peak and tails, that's a great question. I suspect yes, but I don't. I can't say that I've read papers and looked at data that say that for sure. But I kind of, a lot of the invasive species literature goes back to simple diffusion equations, and those also tend to produce a peak and tail. So my guess is yes, but that's a total speculation. I think it's a good question.
Okay. Sam Shiner asks, what does your results say about Deborah Rabinowitz's 7 forms of rarity? Is everything actually rare? Do all of her categories hold up?
Yeah, so my take on the 7 forms of rarity, kind of like the log normal discussion, it depends on your perspective. If you're a macroecologist, all the forms of abundance are correlated with each other: range size, niche breadth or habitat specificity, abundance. They're quite strongly correlated with each other. Studies I've seen, you end up with R's correlation factors of 0.7, 0.8. Now, does that mean that you can't find a dot? So there's this strong central tendency line where abundant is abundant and rare is rare, and the 3 dimensions are not independent or orthogonal. But on the other hand, can you go find an example that's way off that line that has important, Rabinowitz's paradigm has an important conservation implication? Yes, you can. So it's kind of comes down, do you look at the main central tendency line, or do you look at the dots that fall way off the line? And again, you're going to get people who have both those perspectives.
Okay, thank you. Rick Long asks, thank you for the interesting talk. With the increasing anthropogenic impacts on our Earth, how would you see the patterns you exemplify change in our opinion? How can you use these patterns for nature-based solutions?
Yeah, so I guess I would argue that these patterns coming from things like central limit theorems will be general in an equilibrial world, right? So if there were such a thing as an equilibrial human-dominated world, I would argue that you'd find the same patterns. I think it's a more open question how much these patterns are going to be happening in a transitional world where human impacts are, a press environment where the human impacts are strongly changing the world. I didn't show the slide, but I actually there's a nice slide, nice work that looks at how ranges have changed, ranges that have shrunk drastically, how they've changed, and it argues that they all shrink to the edge of the range, contraction to edges, which is an interesting hypothesis. It hasn't gotten the follow-up work that I think that work deserved, but that suggests humans are just sweeping across a range and pushing something to an edge, and that where it's abundant or where it's not abundant really doesn't even matter to the human impact sweeping across. But I think non-equilibrial patterns are a great area for research. I don't know that I think many of the patterns will continue to carry through because they come from central limit theorems, but there's clearly some interesting natural human global change experiments that are happening right now that we should totally be using to look at ranges. Climate change and shifting ranges are a great example. It's our best laboratory for how ranges change through time.
So we're out of time, but I have two more questions, if that's okay with you, Brian.
Sure.
Just a minute longer. Okay. So Gabrielle Mulalay asks, niche breadth and range size seems quite related to. How could this be tested?
Yeah, it's really hard to get a measure of niche breadth that's separate from distributional breadth or distributional extent, because a large range species is going to show up in a lot of habitats, and a small range species is going to show up in fewer habitats. And so it's not a super obvious question to answer, but people have gone through and done that work. The studies I've seen actually show that niche breadth on dimensions like, let's say, diet preference are not strongly correlated with range size, but interestingly, niche position is correlated. So if your niche is something that's a widespread resource, let's say boreal forest, and you like boreal forest, then niche position is more strongly correlated with range size than niche breadth, which is an interesting result. But there are 2 or 3 papers I've seen that kind of did the work to separate size from breadth, and that's what they found.
Okay. All right, thanks. And finally, Ricardo Pernice asks, to what extent do you think the location of habitat boundaries may be responsible for the shape of a species abundance distribution, e.g. symmetric, monotonic, flat, etc.? The location of habitat boundaries responsible for the shape of a species abundance distribution.
Well, I mean, I think the species abundance distribution, which is log normal, comes from many factors. So again, you kind of get this multiplicative process, and I think habitat is a factor, and of course it gets to how sharp is the habitat boundary and how strong is a habitat dependence. If you got a species that won't go where there's no trees and there's a sharp boundary of trees, then it obviously has a very strong effect. But in these more gradual areas, I think it's kind of plays into the central limit theorem as a factor, but not the factor.
Okay. Thank you so much, Brian. These are all the questions. We've managed to answer them all, which is great. Thank you for staying with us a little longer and making sure that we get to answer all the questions. And thank you very much to all of you that have come out for this talk. We really appreciate your participation in our Funk lecture series, and please keep coming. Next month, we'll be meeting with Brett Sheffers, and we very much look forward to his talk as well. So thank you again, Brian, and we'll be in touch.
Thank you.
Bye, everyone.
Bye.